for the next talk is Samir. We have Samir Velankar, who's the head of macromolecules, structure chemistry and bioimaging at the EMBL, European Bioinformatics Institute.
Right. Thank you very much. All right. So first of all, thank you very much for inviting me. Fantastic venue, really amazing organization. So I think this is a model for any conference. Right. Surely I'm not going to let you claim that you don't know enough antibody, because I'm not going to mention antibody except I did twice just now, or have a sketch of that molecule in here. So nothing to do with the topic of this conference. So that's a perfect start. And I just want to not leave this at the end. So I want to thank the team. Those are the resources that we manage. And obviously, none of that can happen without all the funding we get. So thank you to our funders as well. So what I'm going to do in this presentation is to explain AlphaFold, why it works, and what's the impact has been, and where are the challenges in the future. But I'm not going to explain challenge, I'm just going to state them because of the time limit. So let's start in 1980s. So AlphaFold didn't just happen. It's built on many, many years of research and experience. And this paper is from 1986. So why in the first place can we model structures given a sequence? So the first thing to understand is that sequences change more quickly and the structures don't. Structures remain same even if you change sequences as an overall fold. So this is a conclusion of this paper in 1986. So that's the first principle that's important. The second principle that is important is the idea that if we take sequences and if we align them, we might be able to find a position in those sequences where over evolution, the residues are changing in a correlated manner. And what does that mean? The hypothesis is during evolution, you want to conserve the structure. And so if one amino acid changes and the other is changing as a response to that, maybe they are in contact in 3D. And that's the second principle on which all these things nowadays work, which was established in 1994, and so very long time ago. So these are the two things that actually underlie and hold up the whole structure prediction. The other thing that changed after 1990s is we have sequences. So the genome sequencing got us many, many sequences, which means you could have multiple sequence alignments which had enough diversity to actually get better signal for the correlative positions that were changing. Similarly, in the PDB, we got many, many more structures. And the most important thing there with structural genomics was we got more diversity of structures. And so the two things that were important were the diversity of structures in the PDB and the sequence data that we have in the sequence data resources. And both of these are openly accessible. And that's what drives the innovation. But as we drive the innovation, we want to actually evaluate whether we are going in the right way or not. And that's what was offered by CASP. It's a competition that is open to anyone who wants to participate and present their results. And so every 2 years from 1994, there is a CASP meeting. And before that, they give the community sequences and say, predict structures. And those sequences are actually coming from the PDB where we hold the structures until the Cas competition is over. And then you can evaluate the predictions against the experimental data and then say, this person did better than that person, and then it's very objective. And so it's a competition, but it's also collaboration in the sense that at these meetings, everybody is exchanging what worked and what didn't work. And what that means is everybody can go away, learn from what worked, and build on that. And that's what has resulted in the whole AlphaFold. There are many other assessments that happen nowadays for different things. Right, so then that brings us to 2020, where AlphaFold 2, so there was AlphaFold 1 before this, and I'll come to that in a minute. It started predicting structures which were almost equivalent to experimental structures. So the accuracy was fantastic. And these are some quotes from John, who runs CASP, from Demis, who is the CEO of DeepMind. But the important thing here is none of these things. The important thing here is not even the Nature paper. The important thing here is the 64-page appendix that actually explains everything about this method. And that's why the community now has taken this up, and we see OpenFold and all sorts of things happening. It's because the software is open source, but also this treasure trove, which explains exactly what it is doing. And this is really praiseworthy in my opinion. Okay, so I said there was AlphaFold 1, and it was better than everybody else when it presented the results. But AlphaFold 2 has been revolutionary. So what's the difference between AlphaFold 1 and AlphaFold 2? So AlphaFold 1, it followed the traditional thinking, and that was you take the multiple sequence alignments, you try and understand these relationships, and you somehow predict what might be the distances between these residues. And so what you are doing is mimicking an NMR experiment, for example, where you get distance constraints and then put that into a program which understands constraints and can build you a structure. So it wasn't actually building structures. It was only providing constraints that could be helpful in building structures. And that means it did have the limitations of the program that was building the structures, but it also had the limitation that once it actually predicted the constraints, that's it. It couldn't do any better. And that was the information that you could use to predict the structure. And so obviously, it's limiting because depending on how good you are at predicting those constraints, your structure is going to be better or worse. But AlphaFold 2 changed all that. It actually predicts the structure. It goes end to end, and some of the things that are really important is it does not enforce any residue connectivity. It doesn't care that proteins are polymers, and it's important. Why it is important is because it then can balance between the relationships that are short-term in residues that are next to each other versus residues that are further away. And it can adjust them without having to think about, I have to make a polymer out of this. And so it does this for every residue in the protein, and that allows it to kind of build on the understanding. So you could, let's say, start and derive some constraints from the multiple sequence alignment. You, let's say, build a model from that, but that's not protein. That's telling you where the residues are with respect to each other. But it could have information that you could learn from again, and you can recycle this information and slowly have a better understanding of what are the relationship between all the residues with respect to each other. And that's why AlphaFold 2 is better at predicting structure than AlphaFold 1. Okay. So we have predicted structures, but to actually make them useful, you want to also know whether the prediction is any good. And we can't have every time an experimental structure to compare and know whether the prediction is good or not. And in any case, once you have the experimental structure, you don't need the prediction. And so how do you do that? And this is what AlphaFold does better, and this is why AlphaFold structures are useful, because it predicts how good the structures are and its confidence in the prediction itself. So it predicts these two confidence measures. One is, so in a structure, you want to know two things. One is how good the prediction is for individual residues, but that's not good enough. You also want to know how good is the prediction of the relationship between two residues that are in a given structure, because then you know whether the domains are predicted with respect to each other with high confidence or not. And this is what these two metrics do. So let's go over what those are. So the first one is called PLDDTD. So this is the local distance difference test, but this is a predicted value for it by the AlphaFold network. And what it is looking for is how good the packing is locally. And so it's a local predictor. It's telling you how good the residue is it happy in its environment or not? But it's not telling you whether the two residues are placed with high confidence in the structure. And that's what the PLDDT does. And this is derived because of this innovation of not having the polymer in predicting the structure, and individually each residue has its own way of moving about to make the final structure. And the PAE, which is the predicted aligned error, tells you how confident the model is in terms of relationship between two residues. The white part means it doesn't have any confidence. Green means it has high confidence. So how do you use it? You could use PLDDT to, for example, know where the high confidence structures are. So this is all the blue part. The lower confidence ones are different colors. But then the PAE tells you whether this ball actually is predicted with high confidence, where all these different domains are next to each other. And so the way to look at it is, if we take this example, this is coming from here, so individual domains are predicted with very high confidence here. And so obviously, the relationship between residues in a given domain is also predicted with very high confidence. Whereas if you take residues in the two domains, it's in the white region, which means the structure you are getting where you have these domains in particular place is just coincidence. It could be anywhere in 3D. And this is important when you are looking at these structures, and especially for predicting complexes. So this is just one example. This is a protein which I got very early on when we were working on the database. And it has a helix which is sitting in a pore. Now, this is a protein that is exporting things from nucleus. So obviously, we can start speculating that there is this hole and the helix is plugging that hole, and something happens, the helix comes out, and it has all the functional relation, et cetera, et cetera. But before we go there, let's look at the PLDDT and PAE. So the first thing is the PLDDT here is low, which means the confidence in individual residues is low. Secondly, if we look at the PAE with respect to rest of the structure, it's very low confidence, which means it's just a coincidence that that helix is in that place. It has no meaning. The network doesn't think it has done a good job. So no speculations after that. So it saves time as well. Right. So once the AlphaFold was evaluated as part of CASP, the next question is, what do we do with it? So DeepMind approached Emble, and we started working with them. And we had like 3 months to establish a database from scratch. We were experienced, obviously, in the PDB part, which has 200,000 structures now. But we were going to have a database that had about 360,000 structures. But we worked on it. We got it out in time for the Nature publication. And then we have had 3 additional updates. And there was reason for every one of these updates. The first one added all the information on sequences that are in SwissProt. And what that means is these are sequences which are highly studied. There is a lot of literature on it. There is a lot of manual curation on it. And so these are really high value information for which there wasn't enough experimental data on structures. And so that was the reason to do that. The next one was done in a month's time, and this added all the neglected diseases proteomes. And that has, or had, very low coverage in experimental results in the PDB. And so that was the reason behind adding all these proteins. And then we thought, okay, we are going to add more sequences. Which ones should we add? And the question was, why not just do the whole UniProt? And so we added most of the UniProt. So we now have 214 million predicted models in the AlphaFold database. And in my opinion, this is one of the most important data sets we now have after genomics, and it's going to have impact for coming decades. So we are now in an era where there is abundance of structure. Everybody has a structure. If you want one, you have one. You go either to AlphaFold database or you go to Google Colab, you can predict one, right? But that doesn't mean you know how to use it, but you have a structure. So the first question was, this is going to have a huge impact in terms of all these different data resources, different methods predicting structures. How do we make sure that we have a mechanism to know what structures are available? And so we started working on this project called 3D Beacons after AlphaFold 1 because it was quite clear that it is going to become important. The predicted models are going to become important. And so in 2019, we started working on this 3D Beacons project. We brought together all the important data resources, both experimental data resources as well as the predicted models. And we now have a common API that you can use. And so, for example, if you come to the hub, you have an API which looks exactly same as the API here. You ask a question, this is my UniProt accession, do I have structures? It goes to all these, and from wherever there is structure, you get that information instantly. But more importantly, we have all agreed to provide quality metric with these predictions. And so you not only get the availability of structures, you also know how good they are when you get this data. And so have a look at that. Okay, so I have 11 minutes. We're quickly going to see what the impact, et cetera. But in my opinion, in another couple of slides, I can stop if time doesn't permit. So the first impact is, if we look at the PDB, we have about 17% of the human proteome residues. So this is what we have experimentally determined over the last 50 years of PDB. But with AlphaFold, we predicted basically models for everything. And that means you now have almost 93% of residues of human proteome which have structured data in AlphaFold database. So that's just a fantastic start. But in my opinion, the biggest impact, both scientific and social, is the understanding that proteins are not these compact structures that we were used to, because PDB was biased in terms of the experimental methods used to get those structures. And so we had quite a few discussions on whether we should only have the parts that are really properly compact in the AlphaFold database, or we should have the whole protein. And it was consciously decided that we will have everything for two reasons. One, we wanted to obviously differentiate between AlphaFold predictions versus experimental structures in the PDB. But secondly, we now have made the whole protein accessible, because the disordered part here is also a prediction that that bit of the sequence is disordered and doesn't have a structure. So that is information, and that can be used in many different ways, as is said here, and I'll come back to that again in a bit. So how can you use that in structural biology? This is a fantastic tool for all the different sciences. But structural biology can now look at this structure. They can decide what should be the construct by looking at the disordered parts and the ordered parts, and do the experiment more quickly. So that's just a start. There are many other reasons why the structural biologists actually love this and are using it daily. So we have done about 1 billion API requests to AlphaFold database, and this is not people coming to website. This is programmatic use of the information. And we are just going to see amazing things in structural biology. We are going to see, I think, structures in cells. We are going to understand what structures exist, what complexes exist, where they exist in the cell, what are the different conformations that are present in the cell. And this is where structural biology is going. And that's perfect, because structural biology was never about structure, it was about biology. And we want to understand biology, and that's what this is going to do. In terms of how we can use this data, we can look at all, for example, the post-translational modification sites, map them onto the structure like it has happened. So there are many papers here, and this presentation can be distributed. I have no problem. There are many slides that I have hidden. And what we are going to see is new experimental types like proteomics becoming really important for structural biology, because now you can combine cross-linking or HDX or many other techniques with predicted models to answer biological questions. So you don't need to have a high-resolution structure for everything. Depending on your biological question, you can actually use other experimental methods in combination with predicted models and answer your biological question, design new experiments, and test your hypothesis. But in bioinformatics also, there are a huge number of opportunities. So first of all, we have gone from having 200,000 structures to now more than 1 billion structures, if you take the ESM fold into account as well. And so you need tools to deal with this data, to start comparing all these structures to understand evolution, what has happened to a particular function, et cetera, et cetera. So there are huge opportunities there. There are obviously limitations of what we have with AlphaFold. We don't have any ligands, we don't understand any dynamics using it, et cetera. And so there are no complexes, and so there is a huge opportunity there to start predicting complexes, putting in ligands, understanding conformations, et cetera, et cetera. So lots of work still remains to be done. So this is a paper that's coming out next week, actually, in Nature, that we were part of, where we compared all the whole AlphaFold database, everything against everything, clustered it to understand what are the relationships. Because as I said, sequences change, structures don't. And so there are really nice examples here where we have found some immune-related molecules from bacteria and in human, and the structures are very similar. And so there are some histone-related molecules that, again, are common. Some of this is already in the literature, and so now we have structural evidence for that as well. And so have a look at that paper. There is another paper which will be, I think, in the same thing, slightly different way of comparing all these different structures. Okay, so what's happening to CASP now? So CASP has moved on, and we now want to understand complexes and dynamics, obviously. And so this is all the targets from the recent CASP. And as you can see, there are really huge complexes that were predicted using AlphaFold. Most groups used AlphaFold. And then it was really about selecting which is the right model or providing a much better multiple sequence alignment for getting the right prediction. So we are seeing a huge progress in complex predictions as well. Okay, this is, I think, really interesting slide. This is what we usually say is the energy landscape, and protein folding happens where you start somewhere and you get to the structure that is, let's say, minimum energy. And so let's think about, so this paper is really, I think, seminal paper, or will be seminal paper. Because what it is trying to say is, let's take a MSA, multiple sequence alignment, and let's think about that and the energy landscape. And can we link these two things? And the idea here is, if I start with a MSA, that is like starting somewhere on this energy landscape. And so if I modify it, I might start in a different place on that energy landscape. And so if I predict structures by modifying MSAs, do I land in different places with the predictions? And this opens up an exciting opportunity to actually learn dynamics, to actually predict different conformations of the same protein. And this is the idea, and this is the application. And so they did start with different MSAs. So you can take a huge MSA, you can group it in certain ways, and that means you are starting in a different place, and so you get different conformations. So you can see fold switch. Obviously, this is an extreme example, but it's an exciting idea and needs to be explored a bit more. Okay, so what are the challenges? Obviously, we were not used to using structures because they were limited in numbers. And now everybody has a structure. So training, these are still predicted models. And so how do we validate these? How do we know how good they are in terms of experimental data, et cetera? And also making data useful. So the first thing is, how do we train people to actually use these structures? And so we are working on producing material for training, and that will be available hopefully soon on how to use AlphaFold structures. How does this fit into different use cases, etc. One treasure trove that we have is literature. So we have huge amount of information in literature about proteins. We have done lots of experiments, mutation experiments, other experiments, where we have probed the function of that particular protein. And the idea here is we can use that information to actually validate these structures. So let's say there is a mutation experiment. If we can mine literature to get that information, we can map it back to the prediction. And then we can check if the prediction supports the observation. And if it does, then that part of the structure in the prediction is good, validated, and we can use it for something else. And so in my group, we have a project where we're starting to do this work and get information. But how do we make structured data useful? I have 30 seconds. So structures are just numbers and shapes. It only becomes biologically relevant when, let's say, you you expand the symmetry and say this is a complex, but it's still a numbers and shapes. But then you start adding actual biological annotations, and that's when structure actually becomes useful for biological experiments. So you start saying, these are the known variants, these are the binding sites, et cetera, et cetera. So one of those is that we were involved in is getting domains, so assigning domains to understand evolutionary relationship, functional relationships, etc. And so as a whole, our team basically is trying to create a comprehensive system for structured data that includes experimental results, computed models, and I didn't have any time to explain any of this, but we have graph databases which allow you to ask really complicated questions, et cetera, but underlying all this, we're working quite a lot on training. Okay, so last slide. The main messages, open access data, as was highlighted before me as well, is key. CASP was key as well. This is going to change not just structural biology, but biology as a whole, and there are slides after this hidden which have lots of applications. Training is really important, and actually this is going to have amazing impact in the coming years. I also want to advertise a computational structural biology conference if anyone is interested. And these are all the useful resources that we have in my team, and I'll stop there. And sorry, I've gone over.
Okay, that was fantastic. We've moved the schedule around slightly, so you've got a full now until 3:45. We did lose Andrew Ward at the beginning of the meeting, so we're moving two of the shorter talks to tomorrow morning. So that will put us back on schedule tomorrow. So yeah, so we're good for 45 minutes. So enjoy, and then we'll have two more talks, and then we have a half-hour panel, so we can really ask questions and dig into some thoughts about the incredible power of AI.